Hey, what's up YouTube? Welcome to 2.4 of the IB Biology curriculum. Basically, we'll be talking about membranes in this video. So 2.4.1 asks us to draw and label a diagram to show the structure of membranes. Here's my labeled diagram. You got the general phospholipid bilayer. You have the integral protein, and you have the glycoprotein. The glycoprotein, if you'll notice, has the hexagonal rings attached to it, so that's how you know it's a glycoprotein. That's the phospholipid head, phospholipid tails, and then you just draw a little cholesterol thing in there, and you got your phospholipid membrane. Second one is explain how the hydrophobic and hydrophilic properties of phospholipids help to maintain the structure of cell membranes. So whenever it's like these types of explain questions, I start off with something general. So basically, phospholipid bilayer is known as the fluid mosaic model. Phospholipid molecules has a hydrophilic phosphate head. Hydrophilic means that it's polar, it can attach to water, and hydrophobic means it's afraid of water, it doesn't want to get close to it, so the hydrocarbon tails are hydrophobic. In water, a phosphate head will face the water because it's hydrophilic. They want to attach with the water, they're polar with it, and the tails will point to the center and face away from the water, so it'll try to get as far away as possible. And this forms a double phospholipid bilayer. The attraction of the heads to the water and tail with each other makes the membrane very stable. So the head attracting to the water, gives it stability, and the tails here also gives it stability. So it gives a very nice stable structure that you need to be stable. So 2.4.3 asks to list the functions of membrane proteins. I didn't list all of them. I usually just memorize 2 or 3. So membrane proteins, they are hormone binding sites, they can act as enzymes, and they can act as electron carriers. Another easy one is just pumps for active transport. 2.4.4 define diffusion and osmosis. So diffusion is the passive movement of molecules from a high concentration to a region of low concentration, and osmosis is basically diffusion, except it's specific to water, and it's from a high concentration to low concentration. You can also say from a low movement from a low solute concentration to a high solute concentration, whichever one makes you feel more comfortable. 2.4.5 says explain passive transport across membranes by simple diffusion and facilitated diffusion. So simple diffusion literally is simple diffusion. A substance moves between the phospholipid molecules without any help whatsoever. Usually those are lipids, and since it's a phospholipid bilayer, they'll just slide along it and go through. However, some molecules need channel proteins to pass through the membrane. That's facilitated diffusion. Channel protein is specific to one molecule, so only allow a specific type of molecule to pass through. An example is chloride channels, which will allow chloride ions only to pass through and no other molecule. 2.4.6 says explain the role of protein pumps and ATP in active transport across membranes. It's basically start off with definitions whenever it's explained, something general. Active transport is the movement of substances through membranes using ATP. It's useful since molecules can therefore travel against the concentration gradient. So whenever molecules are traveling with their concentration gradient, which is from high region to low region, it doesn't require any ATP. But when you're going from a low region to a high region, for instance, you'll need ATP because you'll need energy because you're moving against the concentration gradient. The protein pumps in the membrane literally pump the substances across or through the membrane using ATP, or ATP obviously is energy form of cells. Each protein pump only pumps certain substances, so the cell still controls what goes in and out. So the cell is still able to retain its power over in terms of which molecules will go through or not. Like any molecule can't just go through the protein pump, it's very specific. 2.4.7 explain how vesicles are used to transport materials within a cell between the rough ER, Golgi, and plasma membrane. So after protein synthesis, proteins are transported to rough ER for modification, transported to Golgi for further modification. Vesicles carrying the protein then fuse with the plasma membrane, expelling the content out of the cell. We'll talk more about that later. It's basically just known as exocytosis. 2.4.8, which is the last learning outcome, describe how the fluidity of the membrane allows it to change shape, break, and reform during endocytosis and exocytosis. So the phospholipid membrane is fluid in nature, as we talked before with the fluid mosaic model. During exocytosis, vesicles will fuse with the membrane, expel their content. So they'll literally just go right up to the membrane, go right through, and let the content leave. During endocytosis, the plasma membrane is pulled inward. So endocytosis probably should be a bit more clear. Exocytosis is when content's leaving the cell. Endocytosis is when it's entering the cell. So the plasma membrane is pulled inward, and a vesicle is pinched off with the molecule. So a large molecule will just approach the membrane, go through, and a vesicle will form around it and get pinched in. In both cases, the membrane reforms to an original state. Because of the fluidity, it's not rigid. It can just, it'll literally just reform together. And both endocytosis and exocytosis are forms of active transport, and therefore they require ATP. And here's unit 2.4. Feel free to just, if you want to copy down notes, pause the video, whatever you want to do. Hopefully this video was helpful. Thanks for watching.